And Dan led the team for the synthesis of this over 1 million base pair chromosome. But it turned out it wasn't going to be as simple in the end, and it sent us back 3 months because we had one error out of over 1 million base pairs in that sequence. So the team developed new debugging software where we could test each synthetic fragment to see if it would grow in a background of a wild-type DNA. And we found that 10 out of the 11 100,000 base pair pieces we synthesized were completely accurate and compatible with a life-forming sequence. We narrowed it down to one fragment, we sequenced it, and found just one base pair had been deleted in an essential gene. So accuracy is essential. There's parts of the genome where it cannot tolerate even a single error. And then there's parts of the genome where we can put in large blocks of DNA, as we did with the watermarks, and it can tolerate all kinds of errors. So it took about 3 months to find that error and repair it. And then early one morning at 6 a.m., we got a text from Dan saying that now the first blue colonies existed. So it's been a long route to get here, 15 years from the beginning. We felt one of the tenets of this field was to make absolutely certain we could distinguish synthetic DNA from natural DNA. Early on, when you're working in a new area of science, you have to think about all the pitfalls and things that could lead you to believe that you had done something when you hadn't, and even worse, leading others to believe it. So we thought the worst problem would be a single molecule contamination of the native chromosome, leading us to believe that we actually had created a synthetic cell when it would have been just a contaminant. So early on, we developed the notion of putting in watermarks in the DNA to absolutely make clear that the DNA was synthetic. In the first chromosome we built in 2008, the 500,000 base pair one, we simply assigned the names of the authors of the chromosome into the genetic code, but it was using just amino acid single letter translations, which leaves out certain letters of the alphabet. So the team actually developed a new code within the code within the code. So it's a new code for interpreting and writing messages in DNA. Now, mathematicians have been hiding and writing messages in the genetic code for a long time, but it's clear they were mathematicians and not biologists, because if you write long messages with the code that the mathematicians developed, it would more than likely lead to new proteins being synthesized with unknown functions. So the code that Mike Montague and the team developed actually puts frequent stop codon, so it's a different alphabet, but allows us to use the entire English alphabet with punctuation and numbers. So there's 4 major watermarks, all over 1,000 base pairs of genetic code. The first one actually contains within it this code for interpreting the rest of the genetic code. So in the remaining information in the watermarks contain the names of, I think it's 46 different authors and key contributors to getting the project to this stage. And we also built in a website address, so that if somebody decodes the code within the code within the code, they can send an email to that address. So it's clearly distinguishable from any other species, having 46 names in it, its own web address, and we added 3 quotations. Because with the first genome, we were criticized for not trying to say something more profound than just signing the work. So we won't give the rest of the code, but we will give the 3 quotations. So the first is to live, to err, to fall, to triumph, and to recreate life out of life. It's a James Joyce quote. The second quotation is, see things not as they are, but as they might be. So it's a quote from the American Prometheus book on Robert Oppenheimer. And the last one is a Richard Feynman quote, "What I cannot build, I cannot understand." So we tried to build a little bit of, because this is as much of a philosophical advance as a technical advance in science, we tried to deal with both the philosophical and the technical side. The last thing I'll say before turning it over to questions is the extensive work that we've done asking for ethical review, pushing the envelope on that side as well as the technical side. This has been broadly discussed in the scientific community, in the policy community, and at the highest levels of the federal government. And even with this announcement, as we did in 2003, that work was funded by the Department of Energy, so the work was reviewed at the level of the White House, trying to decide whether to classify the work or publish it, and they came down on the side of open publication, which is the right approach. We've briefed the White House, we've briefed members of Congress, we've tried to take and push the policy issues in parallel with the scientific advances. So with that, I would like to open it first to the floor for questions. Yes, in the back.
Could you explain in layman's terms how significant a breakthrough this is, please?
Can we explain how significant this is? I'm not sure we're the ones that should be explaining how significant it is. It's significant to us. Perhaps it's a giant philosophical change in how we view life. We actually view it as a baby step in terms of it's taken us 15 years to now to be able to do the experiment we wanted to do 15 years ago on understanding life at its basic level. But we actually believe this is going to be a very powerful set of tools. And we're already starting in numerous avenues to use this tool. We have at the Institute ongoing funding now from NIH in a program with Novartis to try and use these new synthetic DNA tools to perhaps make the flu vaccine that you might get next year. Because instead of taking weeks to months to make these, Dan's team can now make these in less than 24 hours. So when you see how long it took to get an H1N1 vaccine out, we think we can shorten that process quite substantially. In the vaccine area, so Synthetic Genomics and the Institute are forming a new vaccine company because we think these tools can affect vaccines to diseases that haven't been possible to date, things where the viruses rapidly evolve, such with rhinovirus. Wouldn't it be nice to have something that actually blocked common colds? Or more importantly, HIV, where the virus evolves so quickly, the vaccines that are made today can't keep up with those evolutionary changes. Also at Synthetic Genomics, we've been working on major environmental issues. I think this latest oil spill in the Gulf is a reminder. We can't see CO2, we depend on scientific measurements for it, and we see the beginning results of having too much of it. But we can see pre-CO2 now floating on the waters and contaminating the beaches in the Gulf. We need some alternatives for oil. We have a program with ExxonMobil to try and develop new strains of algae that can efficiently capture carbon dioxide from the atmosphere or from concentrated sources, make new hydrocarbons that can go into their refineries to make normal gasoline and diesel fuel out of CO2. Those are just a couple of the approaches and directions that we're taking.